Aspirin, at 125. How a 19th-century synthetic drug continues to shape modern medicine. Few synthetic drugs have remained as clinically relevant as aspirin, acetylsalicylic acid. More than 125 years after its commercial introduction, aspirin remains among the world's most widely prescribed and extensively studied medicines. Combining analgesic, antipyretic, anti-inflammatory, and antithrombotic properties within a single small molecule, aspirin has transformed medical practice. It remains a cornerstone of cardiovascular therapy, while gaining renewed attention for applications in oncology, infectious diseases, and immunology. The story of aspirin began long before modern chemistry. Historical records from ancient Egypt, Mesopotamia, Greece, and China describe the medicinal use of willow, Salix species, bark to relieve pain, fever, and inflammation. Around the 5th century BCE, Hippocrates recommended willow preparations for pain and childbirth-related fever. The active constituent, salicin, was isolated in the early 19th century, and subsequent chemical studies showed that its metabolite, salicylic acid, possessed potent anti-inflammatory activity. However, salicylic acid frequently caused severe gastric irritation, limiting its therapeutic use. The breakthrough came in 1897, when chemist Felix Hoffmann, at Bayer, chemically acetylated salicylic acid to produce highly pure and stable acetylsalicylic acid. Bayer patented the compound and introduced it commercially under the trademark Aspirin in 1899, making it one of the earliest successful synthetic pharmaceuticals. Initially marketed as an analgesic and antipyretic, aspirin rapidly became a global therapeutic standard because of its improved tolerability and efficacy compared with salicylic acid. Today, aspirin is available in numerous formulations, including conventional, chewable, enteric-coated, effervescent, intravenous, and buccal preparations. Aspirin's remarkable versatility arises from its unique pharmacological mechanism. Unlike other non-steroidal anti-inflammatory drugs, NSAIDs, aspirin irreversibly acetylates cyclooxygenase 1, COX1, and cyclooxygenase 2, COX2, thereby inhibiting prostaglandin and thromboxane synthesis. Suppression of prostaglandins produces analgesic, antipyretic, and anti-inflammatory effects, whereas irreversible inhibition of platelet COX1 prevents thromboxane A2 formation, suppressing platelet aggregation throughout the platelet lifespan. This irreversible antiplatelet action distinguishes aspirin from most NSAIDs and underlies its exceptional value in cardiovascular medicine. Low-dose aspirin, 75 to 100 milligrams per day, remains the gold standard for secondary prevention of myocardial infarction, ischemic stroke, and other atherosclerotic cardiovascular diseases. Decades of randomized clinical trials have demonstrated substantial reductions in recurrent thrombotic events among high-risk patients. Although routine use for primary prevention has become more selective because of bleeding risk, aspirin remains indispensable for carefully selected patients with established cardiovascular disease. Beyond cardiovascular medicine, aspirin continues to find new therapeutic applications. Growing evidence indicates that aspirin modulates inflammatory signaling pathways, including inhibition of nuclear factor kappa-B, ANF-kappa-B, regulation of immune cell activation, and production of specialized pro-resolving lipid mediators such as aspirin-triggered lipoxins and resolvins. These actions have stimulated investigation into aspirin as an adjunctive therapy for systemic inflammatory disorders, including sepsis, acute respiratory distress syndrome, ARDS, recurrent pericarditis, Kawasaki disease, rheumatic fever, and adult-onset Still's disease. Aspirin has also attracted considerable attention in oncology. Long-term observational studies and several clinical trials suggest that regular low-dose aspirin may reduce the incidence of colorectal adenomas and colorectal cancer while potentially limiting metastatic progression through inhibition of platelet-mediated tumor dissemination. Although evidence for cancer prevention remains strongest for colorectal malignancies, ongoing clinical studies are evaluating its role against several other cancers. However, benefits appear to vary among patient populations, emphasizing the need for personalized treatment strategies. Another emerging area is infectious disease. Experimental studies have demonstrated antiviral activity against both RNA and DNA viruses, primarily through modulation of host inflammatory pathways rather than direct viral inhibition. During the COVID-19 pandemic, aspirin received renewed interest because its combined anti-inflammatory, antithrombotic, and potential antiviral properties could theoretically mitigate hyperinflammation and thrombotic complications. While clinical outcomes have been mixed, aspirin continues to be investigated as an adjunct therapy for viral infections, HIV-associated inflammation, influenza, and immune-mediated complications. Despite its extraordinary success, aspirin is not without risk. Gastrointestinal irritation, peptic ulceration, and bleeding remain its principal adverse effects, particularly during prolonged therapy or high-dose administration. Intracranial hemorrhage, although uncommon, represents the most serious complication. Aspirin-induced hypersensitivity, bronchospasm, and aspirin-exacerbated respiratory disease occur in susceptible individuals. The drug is contraindicated in children with viral illnesses because of the risk of Reyes syndrome and should generally be avoided during the third trimester of pregnancy. Co-administration of proton pump inhibitors can substantially reduce gastrointestinal complications in high-risk patients, while individualized risk-benefit assessment remains essential before initiating long-term therapy. More than 2 decades into the genomic and precision medicine era, aspirin continues to evolve rather than fade into history. Advances in molecular pharmacology, platelet biology, inflammation research, and cancer immunology continue to reveal previously unrecognized mechanisms of action. Ongoing investigations into precision dosing, pharmacogenomics, combination therapies, antiviral activity, neuroprotection, and cancer prevention may further expand its clinical utility. Over 125 years after its introduction, aspirin remains a remarkable example of how a simple synthetic molecule can continually adapt to modern medicine, demonstrating that even the oldest drugs can yield new therapeutic opportunities through advances in biomedical science.